Good evening, ladies and gentlemen. Welcome to Didsbury Sidebar. Thank you for coming. We are raring to go. This evening, our speaker is Dr. Niamh Fox from Manchester University, and she's going to talk to us about polymers and what happens to them when you stretch them. Thank you, Niamh.
Cheers, thank you. Hi, everyone. My name is Niamh. I'm going to talk to you today about one of my favorite topics, which is polymer mechanochemistry. I am happy for you to shout up with any questions at any point. If there is something that I have said that seems ridiculous or unclear, please do stop me at any time. Don't have to feel like you have to wait till the end. And yeah, I am a lecturer in Polymer Science at the University of Manchester. I joined the Department of Materials about a year ago, and I research this area of polymer mechanochemistry, which I'm going to talk to you about this evening. The first thing I have to do is put up a little bit of a caution. I sent the slides for this talk to a pal of mine, and she said, there is far too many chemical structures in there. Take them all out. I didn't take them all out. I have to admit, there are still some in there, still quite a lot in there. So imagine what it was like before I sent them to her. But we all have Beth to thank for how few there now is. And I promise that the ones that are in there are not particularly important. I'm not going to dwell on a lot of the chemistry. We're going to talk about concepts, but some of them are there just to illustrate some of the points that I have that I'm going to talk about. And the other point of caution that I have to start this off is I may require a small bit of audience participation. We'll see how it works. But so the caution is out of the way. Bear with me. We'll see a bit of chemistry, a bit of audience participation, and a lot of polymer mechanochemistry. So the way I'm going to structure this talk, and it's very loose in terms of a structure, is a bit of talking about past, present, and the future of polymer mechanochemistry. In case you can't guess, that is, of course, me on the far right playing with some Lego. Destined to be some kind of future engineer or material scientist as a child. So I'll talk to you a little bit about how I got here, my journey into mechanochemistry. Then I'll introduce this idea of polymer mechanochemistry, what it is, what it can be used for, and then at the end, talk a little bit about where I think it might be useful in the future. And then if you have any suggestions of where you think it might be useful in the future, that will be even more interesting to me than listening to what I'm suggesting at the end. Okay, so talking about how did I get here, you will be, I hope, very unsurprised to hear that I am Irish, both with an accent and a name like Niamh. I am from Dublin. I grew up in Dublin near the Phoenix Park, and you absolutely cannot see there the map of Dublin, but there's a large park there, and I grew up beside it. I spent all of my life in the one spot. I then grew up in this village, which is called Chapel Isid. It's a little bit similar both in distance and aesthetic, maybe to Didsbury versus Manchester, that kind of distance outside of Dublin, on a river, really super place to grow up. Then when I finished school, I went to Trinity College in Dublin, where I studied chemistry. I ended up going to Trinity, if you listen to my parents, because it only took one bus to get there, as opposed to the other university in Dublin, which took two buses to get there. But yeah, that was a fantastic place to be. I studied chemistry. I was absolutely fascinated by making up different solutions of different colors and things. And you'll see lots of color throughout this talk. And you can see that that is something that really catches my eye. So I studied chemistry. Like I said, I grew up in Dublin. I went to university in Dublin. I was kind of itching at this point to get out somewhere a little bit further away. So as an undergraduate, I spent some time at the University of California in Santa Barbara, which is an absolutely fantastic university. This is a picture of its front gate. What you can see maybe the most striking bit for me was university was literally right beside the beach. You didn't even have to cross a road to make it to the beach from the university. I didn't understand how anyone did a single piece of work. But it was a fantastic institution. There were several Nobel Prize winners on staff while I was there. They still did manage to do work somehow. And I could understand how I could do work there because, again, as you may be able to guess, I am not the sort of person who spends a lot of time at the beach. So I spent a reasonable amount of time in the lab. And what I was doing was looking at something that maybe doesn't sound super interesting at the start. What I was looking at was how we could measure and replicate surface roughness. And that, again, doesn't sound particularly exciting. So we have a rough surface, so what? But surface roughness is something that's really important for adhesion. And the group that I was in was looking at this because they were looking to mimic this phenomenon, which is the hierarchical structure of gecko's feet. So because of the surface roughness of gecko's feet, they have this ability to be able to walk up walls. And this is because they have lots and lots of different fibers on the base of their feet that creates a really large surface area and allows them to have this contact with the wall. And this is all due to their surface roughness. And being able to measure and replicate that was something that they were super interested in terms of adhesion. And not to be outdone, at a lab in California, looking at the surface roughness of gecko's feet, they did have a gecko in the lab. The gecko's name was Dude. He was definitely the most popular member of the lab. But it was quite a fantastic place to spend a bit of time. And looking, again, starting to get this idea and feel for research. I came back to Trinity then and did a PhD, particularly looking at fluorescent polymers. Again, like I mentioned, quite interested in color, changing color, how we can control color. And I was interested in these fluorescent polymers for things like LEDs and sensors. So again, I got away from Dublin for a little bit. I got pulled back in. And then I wanted to explore a little bit further. So I went to MIT, Massachusetts Institute for Technology in Boston. And I spent about 3/4 of a year there. I had a fantastic time. I got to see every type of weather that existed in Boston. There's a joke in Boston that if you don't like the weather, you should just wait an hour. You'll get everything that you could want in one day. I had a really super time. I don't have any photos of the research that I was doing, again, looking at these conjugated polymers. The group that I was in had made quite a big name for themselves, developing these fluorescent polymers for use as detectors for explosives. So quite a cool kind of piece of research and technology. I was looking at much more benign things. They didn't let me near any explosives as a kind of visiting PhD student. Actually, I'll be very honest, there was no explosives in the lab at all. Sorry, just to put everyone's mind at ease. Nobody was, but we were developing lots of cool sensors using these conjugated polymers. And then this was all kind of set in chemistry departments. So I spent a lot of time doing chemistry and looking at the chemistry of these materials, but I was interested in what happens then in the next step. So someone develops some exciting new polymer in a chemistry lab, but how does it go beyond the lab? What happens when it gets processed and what happens when it gets used? So I moved, after finishing my PhD at Trinity, I moved to the engineering department at the University of Cambridge. And I started looking at what happens when we start to try and deposit these polymers into an application. What happens when we try to print them? And that is where my interest in this polymer mechanochemistry started. We're going to talk about a lot later on, so I won't focus on now. But then, just to say, moving on, I really enjoyed this idea of engineering, trying to put these materials into practical application. So I stayed on to do another postdoc, again in the engineering department at Cambridge, this time looking at what happens to materials when we try to process them, particularly for technologies for delivering drugs to hard to treat cancers. So cancers in difficult places in the body where it's hard to deliver drugs, what happens when we develop new technologies to deliver those drugs and how the processing of the devices alter their function. So I spent 4 years doing that, which was something incredibly new for me because I did no biology as a student. So it was quite a steep learning curve, but something really fun and interesting and incredibly important And it's been a really exciting project to see how it's developed and where it's gone. So all of that is kind of a very long way to say how then I ended up in the materials department. It's kind of a mixture between the chemistry where I started, the engineering where I spent a long time, and then material science feels like the perfect marriage of the two of those. So I get to look at the chemistry, the engineering, all kind of through this lens of material science. And what we're going to talk about, or what we're going to focus on in terms of material science, is this idea of polymer mechanochemistry. So what happens when we deform and strain these particular types of polymer materials? And I hope this is going to play. Is it going to play? Yes, it is. So we have a material here that is a polymer film containing what is known as a mechanophore. We're going to talk about that later. And when we stress and strain it, it changes its chemical properties, and we get actually it gives off light, so we get a change in its fluorescence. Okay, and we're going to talk about how that happens, why that happens, and why it might be useful. So this comes on to our first question to this audience that I know has people who know the answer to this. My first question to the audience is, what is a polymer? I told you there would be audience participation. There was a big caution at the start.
A long series of monomers.
A long series of monomers, absolutely spot on. I will follow up with what is a monomer. Okay, yes, so we've got a lot of different components that in that single, well, kind of two-parter answer. It's a long chain, absolutely. So a polymer is a long chain of molecules. It's made up of repeat units of monomers that are joined together. So we have a sequence of repeat units or monomers that are joined together to form this really long chain. And the thing about polymers is they are a really long chain. We're not talking about small molecules. We're talking about something that's really, really big. And the last point that was mentioned in that answer there is that it has something to do with plastics, right? So effectively, we have this scenario where all plastics are polymers, but with the quick caveat that not all polymers are necessarily plastics, okay? So small distinction, but plastics is generally what we can think of. So what is a polymer? We know it's this long chain, it's got these repeat units, and it is used in lots of applications, in particular plastics. So we have a long chain. And a lot of the time when we think about this long chain, we think about a piece of string. So I brought a piece of string. Did I bring a piece of string? I did. Brought a piece of string. So we have a really long piece of string, right? Coils, it changes its shape, it's really flexible. But the other thing to remember is that it's not just a piece of string, it is also made up of all these monomer units. So it has a lot of repeat units of the same thing. This all is essentially just one piece of string. It looks like one thing. But when we think about a polymer, it's made up of lots of monomers joined together. So actually, rather than think of maybe like a piece of string, we should think maybe more like a chain of paper clips. Oh, you can tell I was really busy this afternoon at work. Again, we have this flexibility, but what we have is these monomers all joined together. So we have lots and lots of paper clips, same kind of movement. It's really flexible, really long chain. But we still have the same idea. It's this long piece of material that can coil and change and be flexible. And we have lots of different types of polymers. So we've mentioned plastics, but we have a lot of natural polymers. So DNA is a polymer. It's a really long chain piece of material that's made up of repeat units, in this case, nucleobases. We have natural rubber, which is something that can essentially be farmed, is not quite the right material word, but it can be extracted from the rubber tree, and then we can essentially process it to make rubbers that we use in all kinds of different applications, from our tires to different nitrile gloves, like stuff like this that we'd use in the lab. So we have these natural materials that we can use, and then also we have a whole ton of synthetic materials. So we can think of all the kinds of plastics that we use in our lives, from plastic bottles to the nylon in tights, etc. I could put any number of applications on this slide for the use of synthetic polymers. So kind of polymers are ubiquitous in our lives. We use them all over the place, but that wasn't necessarily always the case. And actually the history of polymers, I'm always kind of amazed to think about is a little bit shorter than I would expect. Polymers have been on the scene for, recognized to be on the scene for maybe about 100 years. So in the 1920s, this guy here, Hermann Staudinger, said that polymerization was linking of small molecules together to form high molecular weights. So that's actually the answer that we came up with. Here, when I asked, what is a polymer? That's what people said. But in the 1920s, when Hermann Staudinger said this, people said, you are absolutely ludicrous. There is no such thing as a really long molecule. That is effectively just, you haven't purified your product correctly. They're all just small molecules. There is no such thing as these long chain polymers. So he actually got a letter from a Nobel laureate, a friend of his, who said, essentially, please stop banging on about this polymer stuff. It's not real. There's only small molecules. Big, long molecules don't exist. Thankfully, Staudinger was both very good at his job and he didn't quit easily, so he kept going and he was proved right. He essentially started this field of polymer science and the idea of these really long molecules. And he was also the first person who started to think about what happens when we deform or try to degrade these really long molecules with mechanical force. So he is both the kind of father of polymer science and also polymer mechanochemistry in one kind of fell swoop. So before the 1920s, people were using polymer materials like cellulose and all kinds of different things for practical applications, but they didn't know that polymers had these long chain structures that had a very high molecular weight. So then, just to really hammer home this message, quick question for you to think about: how many of you have used some kind of plastic today? Oh. Absolutely every single person will have used some kind of plastic. So I have some examples here. You won't necessarily have used all of these today, but they are literally everywhere we can think of. We store our food in them. We use them as children's toys. We have them in our contact lenses. We have them in our kitchen utensils. We have, really importantly in Manchester, they're part of what makes our clothes waterproof. We need them everywhere and anywhere. They're in sports equipment. They're in also liquid formulations. So they're not just necessarily solids. We can have them as viscosity modifiers and things like paints and sun creams. They are absolutely everywhere. So polymers are really, really important. And I think because of their association with plastics, sometimes they get a bit of a bad rep. But I hope you'll agree by the end of this that that's not necessarily the case. Sometimes they can be useful. A lot of the times they can be useful. We'd be really, really stuck if all of these things all of a sudden were removed from our lives. We need polymers and polymer science as we go forward. So this comes back to this question then. We need them. Why are they so useful? What are the aspects of polymers that really make them such a wonderful material when we compare them to something like a small molecule? And when we're thinking about this, I want to have comparison. Like I said, we have our long chain of paper clips, our very flexible long chain material, or you can think of it like a plate of cooked spaghetti, okay? Both of those analogies hold true when we're talking about polymers. So the first thing is they're really low density. So compared to something like a metal, we can make a much, much lighter part when we use polymer or plastic compared to if we made it out of metal. And that's incredibly useful if we're thinking about things like transport, where if we can make the vehicle lighter, we need less fuel to get moving. So they're low density. They're flexible. We have this movement in the polymer chain, not only just in the chain, but also in the material. We can bend and flex the material without breaking it necessarily. They're easy to process, and that was kind of one of the first things that, like I say, got me interested in this idea of polymer mechanochemistry, was how truly easy are they to process. So can we can melt them and shape them into the shapes that we want. We can then, depending on what type of plastic they are, we can remelt them and remold them into a new shape and make something useful again. and we can do that process a number of times. They're reasonably chemically stable. This is not a blanket, they are stable, I will not say that, but they have a reasonable chemical stability. They're chemically versatile, so by changing the chemical structure of the backbone, we get a change in their properties. So we can think about a whole range of things that we can synthesize with a range of different properties, depending on their structure of the polymer. And they have a really high mechanical strength, again, when we think about in comparison to other materials and in terms of their density and flexibility. So where does that mechanical strength come from? And part of it, again, comes back to this the long chain nature of polymers. Lost one of, oh no, I have lost one of my props. And part of that is because these polymer chains can entangle. So this is a number of just bits of string, cut them all up and essentially just scrunch them into a ball. And what we can see is that they've started to entangle. So we have polymer chains crisscrossing because of their length in nature. And essentially, if I was to pick up one bit of it, it picks up all the rest of it because everything is entangled and forms this structure that's enmeshed. And we have polymer chains looped around each other. And that gives us the solid nature of the material. So we can think of these polymer materials, the importance of their really long chains, their strength, and their easy, the possibility of processing them quite easily. And this mechanical strength and their easy processability is what really appealed to me when I started thinking about polymer chains. And then what happens during processing is that we effectively put them under mechanical force. And that's what led to thinking about this idea of polymer mechanochemistry, which is not mine. I mean, that's where I got interested in it. So that leads nicely. We've talked about the kind of past where my motivations for coming to this topic have come from. And now let's start looking a bit more in depth into this idea of mechanochemistry. So we're going to have an intro to mechanochemistry, look at some of the applications, and then some of the work that I've been doing recently. So the first thing is we've done very well at what is a polymer. You can ask very quickly, what do you think mechanochemistry is? Yeah.
The application of physical force.
Absolutely, yes. So it is using mechanical force to drive a chemical reaction. So quite classically, we have this idea in chemistry of using things like light or heat or electricity to drive chemical reactions. But mechanochemistry is the study of using mechanical energy or mechanical force to drive chemical reactions. And if you look in the polymer mechanochemistry literature, that would suggest that the field is not quite that old, but actually the ancient Greeks used mechanochemistry when they ground things together to make things like cinnabar. So we knew about grinding materials up together to change their chemical properties thousands of years ago. Michael Faraday did some mechanochemical experiments, but Michael Faraday did every experiment you could ever imagine, so that's not necessarily that exciting. But Michael Faraday looked at changing the halide salts in silver compounds, again by grinding. We all may or may not have, I certainly had one of these types of shoes where if you stamp on them, you light up the base of them. That's a bit of mechanochemistry. You're changing the crystal structure in a piezo that's in the shoe, and that causes the light to light up a little bit. And we all may have done lots of this during the pandemic, making bread. When you stretch out the dough when you're making bread, you essentially change the structure of the proteins in it and you activate the gluten when you're kneading your bread. So we all can do bits of mechanochemistry in all kinds of parts of our life, okay? So mechanochemistry is something that maybe we don't talk about that much, but we do a lot of it, okay? So that's just mechanochemistry. But we now need to think about what happens when we're thinking about how this affects polymer chains. So we said that polymer chains are these long, flexible materials. They tend to coil up and form a kind of very random coiled structure. And we can think about what happens when we stretch an elastic band. So essentially, when we stretch an elastic band, I would give these out to everyone to have a go at doing some mechanochemistry, but I have to say I have been teaching the MSc students this week and given them all, so I have no idea where these elastic bands have been, and I don't want to pass this on to anyone else. But when we think about stretching an elastic band, essentially what we're doing when we pull this elastic band is uncoiling those polymer chains, okay? So when we have an elastic, when it's at rest, the polymer chains are in their coiled state. And when we start to put some force on them and pull the elastic band, we start to uncoil those polymer chains and start to pull them out. So if we think about, again, coming back to our paper clips, this isn't great, but you can see we have effectively an uncoiled random bit of our polymer. And then when we start to pull it out, we start to stretch out that coil and we start to stretch the polymer chain. And that's what happens when we're stretching this elastic band. So we're at rest. If we look, imagine this mess that I have drawn is a polymer chain, a random coil. And then when we stretch it, we start to stretch that coil, we start to elongate parts of it, and we start to stretch them out. And then when we release the force, it goes back to its random coil state, okay? So we go from a random coil state to something that's stretched and aligned, and then if we release it, we go back to this random coil. And where polymer, this in and of itself isn't polymer mechanochemistry, we're changing the structure, yes, but it goes back to what it started as after that. Where the exciting things happen is what happens if we really, really stretch this? What happens if we really pull on the polymer chain? What can we get it to do? And that's where this field of polymer mechanochemistry comes in. What happens if we really, really, really stretch out these polymer chains? And the answer is if we pull them far enough and fast enough, essentially we can cause breaking of the chemical bonds in the polymer. And that's not great, because in reality, what we want from the polymer is its long chain nature. We want it to coil and form these kind of strong structures where we have these entanglements. If we all of a sudden pull on these chains and we break them, our polymer is now half the length it used to be, and then it's almost half as useful, right? So we've reduced our molecular weight by half, and we really don't want that. And if we can think about that again, we look at our polymer. I'll go back to the thread one if I can get you to. Okay. So if we have our random coil, we start to pull it out and pull it out and pull it out and pull it out. If we really pull it, what we can see is that tension builds up along this thread, and actually where the tension is going to be highest is at the middle point of this thread here. It's not going to be that high here. It's going to be all the way in the middle where we pulled it really, really far. And this is where the bond is likely to break, okay, if we stretch out our polymer chain like that. So if we pull them far enough, fast enough, with enough strength, we can cause these bonds to break. And what's really important about pulling these chains is the chain length. So if I was to just pull one, do I have a smaller chain? I don't have a smaller chain. So if I had just a very short polymer chain and I was to pull, I could never build up enough tension here to break the bond in the polymer chain. I need the length of the polymer chain to be able to build up enough tension, because I'm transferring the tension through the bonds in the backbone. So I need stuff that's super, super long to be able to, I couldn't do this on my own. Yeah. So chain length is really, really important. And we can think of this like we were trying to stretch out penne versus a big long piece of spaghetti. It's easier to break the spaghetti because we can build up enough tension when we pull it out versus trying to break the penne, okay? So chain length is really important when we're starting to think about polymer mechanochemistry. So then the question is polymer mechanochemistry a bad thing? So I've just said that we like polymers because we have these really long chains. They give us strength, they give us flexibility, they give us low density. And the really important thing is how long they are and how they can entangle. Then I've said if we pull them really hard and really fast, we break them, and that reduces their molecular weight. So is my whole career just a really bad choice? Obviously, I don't think so. So the idea is, yes, we get the breaking of these polymer chains, but about 15, 20 years ago, actually even before that, there was this move to try and see, okay, what if we can turn something that is mostly destructive and actually use it as an input to do something productive? So rather than just having a polymer chain where we break a random bond in the middle of that chain under tension, what if we put a chemical species in there that does something productive when we put it under tension? So what if we put in something that we now call a mechanophore that undergoes a chemical change that we would know and expect under force? And that's this concept of the mechanophore. So I have sample mechanophore here. This is one of the chemical structures that I refused to take out. Apologies. So this is spiropyrin. And in its unstressed state at rest, it's clear. But if we start to pull on the chains on either side here, you see the squiggles, there are polymer chains. If we start to pull on them, we can break open the carbon-oxygen bond, which is shown in red here. And we move to a scenario where we have this species here, which is known as myrcene, which is purple. So we can go from something that is clear to something that is purple under tension when we break the bond that we expect in the polymer chain. Okay, so there's a couple of things that we need to think about when we're designing these mechanophores to do something useful for us. We need to include a weak bond. We'll get onto that in a second. We need to make sure that we have a long enough chain in our polymer, because like we said, if we have a short chain, we can't build up enough tension to break the bond. So we need a long enough chain to build up enough force. And we need to have the correct anchoring points. So if we were to add our polymer chains to different points in that chemical structure, we would not direct the force along the carbon-oxygen bond that we need to break. And therefore, we wouldn't get activation of our mechanophore. Okay, so we wouldn't go from this clear, colorless state to the purple myrcene form. So let's think about these weak bonds. When we first stretch this out, you can see that essentially everything in this polymer chain is the same. So we have all of the monomers are the same in this backbone, and effectively we'll get random scissions somewhere in the middle of the chain where this tension breaks up. But if we have a mechanophore, we've now included a weakened bond and one that we have programmed to go. So I have represented that, but now I have a paper clip in the middle of my polymer chain, okay? So this may or may not work. I have not tested this one before. I'm gonna ask, and we're gonna try and pull these and see what happens. What happens? Oh, our bond breaks. So not only did it broke at the point of our paper clip. So we directed the tension along the polymer ribbon and effectively pulled open our mechanophore that ripped as we put it under tension, okay? So we didn't rip some point randomly on the rest of our chain. It ripped at the point where we have this weakened bond. I didn't actually expect the knots to hold. That was why I was worried about this. And then we have a little bit of a thought experiment, because something that I get asked a lot then is, okay, but how do you know which is the bond that's going to break? How do you know where in the paper clip is going to break? And the reality is that we need to align the bond in the chemical structure with the axis that we're pulling on. So if we're pulling in this direction, we need our bond to align with that, because we essentially need to pull open our bond. So if we think about moving from the idea that we just had where we had our paper clip in line with the ribbon, what if we now think about pulling it like this? I have not attempted to make this because I know that if I put knots here, they'll just slide. But if we think about this conceptually, if we start to pull from the sides of the paper clip, we're effectively not going to open it because we don't have programmed weakened bond, which is where the paper clip opens up on its edge, okay? So the anchoring points are really important for our mechanophore. So we know that we can put the tension across the correct bonds. And then we can do control experiments to look at these to see, okay, is this really mechanochemistry? How do we know that it's not just light or heat or electricity that's driving these things? So first, if we have our mechanophore that's linked in the center of the polymer chain, with the weakened bond aligned to the pulling force, we should get activation of our mechanophore. So we should see our purple color in this case. Then if we do something like put it at the end of the polymer chain, so again, we can now use this here. If we put this here, we pull, we're not going to activate our mechanophore because we're not going to get enough tension in this portion of the chain. The tension is highest in the middle. So we're not going to activate our mechanophore in this case. If we then just have a mixture, so if I was to have, you know, my random polymer foil and my paper clip, we're not going to get activation of this mechanochemically here because, again, we can't put any force over this if it's not connected into our polymer chain. So if we have this covalent mixture and we see no activation, then we have a good idea that we would get that our activation is mechanochemical when it's in the middle of the chain. And then finally, if we have this idea of a dysfunctional linker. So again, in this case, if I was to attach the polymer chains to the middle of the paper clip, or in this case, if I was to have one at one end and one here, again, we wouldn't build up tension across the correct bond in the mechanophore to activate it. So we can do all of these control experiments. So, well, the top left is the positive control. We expect to see activation in that point. And then in all of the others, we expect not to see activation. And from those two, from this combination, we can tell that we do have mechanochemical activation of our mechanophore. And just looking at a comparison of what we mean by weak bond. So if we're trying to break covalent bonds in the backbone of a polymer with no mechanophore, they can be on the order of tens of nanonewtons, which is absolutely tiny and almost impossible to think about what kind of force that is. But mechanophores are almost an order of magnitude lower than that. So we have bonds that are 10 times weaker, that are easier to break open. If we think then about unfolding proteins, this is another order of 2 of magnitude lower than that. And if we think about the force required to unzip DNA, we're down at 10 piconewtons. So we have this whole range of different kinds of bonds that we can think about trying to manipulate mechanically, okay? And all to do with this mechanochemistry involving polymer materials. So then, this one, you're just going to have to bear with me. This is not even this, but this is an absolute overload of chemical structures. And I just want to show you that there are an absolute range of different types of mechanophores that have been developed over the last 20, 15 years, okay? I do not expect you to be able to see any of these. I just want to show you that there are a range of different types of mechanophores that have been developed. They do a whole range of different things. And it's really exciting to think about where they might go next and what people might do with them. So there's whole tons and tons of chemistry being developed by people far greater in the lab than me. Okay. So then if we come back to this video that we saw at the start of stretching this polymer film and getting a change in the fluorescence, what we have is a mechanophore that looks a little bit like this. So it is rotaxane. So we have essentially an axle. We have a macrocycle bound around it. And as we move the macrocycle, we change the ability of the macrocycle to quench the fluorescence. And as we turn on and off the fluorescence, we can see it as we stretch the polymer chain. Okay, so as we shuffle this ring component back and forward across the axle, it turns on and off the fluorescence of the material, and we can see that in our film as we stretch it on the macro scale. Something that's happening essentially on the molecular level then gives us this fluorescence on the macro scale. Okay. So that's a very whirlwind introduction into polymer mechanochemistry and what it is. And we're going to just take a very brief look then at what people are starting to think about using it for. And there are a whole range of things that people are thinking about using it for. It's incredibly useful when we start thinking about how we characterize materials. So it can tell us things, like I say, that are happening on the molecular level that we would never be able to see. So we can use it to understand the characteristics of materials, how they fracture and behave when they're deformed. It can be used to tell us how materials used for things like soft robotics, how they're doing, when they might fracture and fail, how useful they are. That's not just soft robotics, it's any kind of plastics. We can think about using them as sensors for telling us when materials are degrading. We can think about using it for artificial muscles. So this is a really exciting example where if you stretch a polymer in a gel network and cause it to break, you've broken that bond. But if you have materials to reform the polymer in that gel, you can get it to build back stronger. And that's kind of the same principle that muscles work on. So we stretch out the polymer chains or the proteins or whatever that's in our muscles. If we cause them to break, we reform them and build back stronger. So that's really exciting. We can think about controlled degradation. That's something we'll talk about a little bit more. Particularly useful for self-healing materials. So again, if we've broken that polymer chain, how can we think about rebuilding it? And then finally, drug delivery. So we're going to look at drug delivery first. And there's a lot of different ways that we can use polymer mechanochemistry in terms of drug delivery. The first is we can think about having, oh, lost it again. Okay, we can think of having our mechanophore being the active drug that we want to deliver. So if our mechanophore is the drug that we want to deliver, and we apply some force, we can then release the active drug at the site that we want. We can have it encapsulated in some kind of polymer structure. And when we pull on the polymer structure and unravel that, we release the active drug. Or we can have it attached to different polymer nanoparticles. And again, as we pull them apart, we release the active drug. So in each of these cases, we have something that is inactive and not useful in the body until we start to trigger polymer mechanochemistry, and that releases an active drug. And why might that be useful? If we think of having to deliver something systemically, it goes all through our body. We don't necessarily want to have the active drug floating free everywhere and anywhere. But if we have the inactive material, the polymer mechanochemistry component, floating around, and then we were to apply something like ultrasound at a particular place, we can cause the polymer chains to stretch out and then release our active drug at the point where we apply our ultrasound. Okay, so that's super exciting. And lots of different materials have been shown using this method here. Things like cancer treatments and all kinds of different small molecule drugs have been released using this polymer mechanochemistry. And then the other thing that we can think about is looking at controlled degradation. So what we're very conscious of is, we've mentioned that polymers are used an awful lot, or polymers are synonymous with plastics. And as we know, we want to think about how we can deal with plastics at their end of life, how we can essentially recycle them. And mechanochemistry might be able to help us here. So if we start to think about building up these long polymer chains, we could be able to use mechanical force to pull them apart at the end and then reprocess them. So they have these implications for sustainability. This was one of the first forays into trying to use polymer mechanochemistry for recycling purposes. It had some really stringent chemical conditions on how it could be used to recycle, so not very applicable across the board. But that was about 10 years ago. And then recently this year, ever since then, there have been strides to try and make polymer mechanochemistry useful in terms of recycling. And we have this idea where we can include these mechanically active bits into the polymer backbone. And when we apply mechanical force, the polymer doesn't necessarily break down, but it changes its chemical structure enough that it can be broken down further with things like treatment by hydrolysis. And we get back to our original starting material that we can reprocess and make into something new, which I think is incredibly exciting. So that was a very brief touch on some of the applications of mechanochemistry. And then the last bit I'll talk about in terms of the actual research and what's going on is kind of where does any of my research come into in any of this? And what I'm looking at is what happens to polymer mechanochemistry when we look at different scales. So far, we've really been looking at kind of single chains, what happens conceptually when we pull out a single chain. But I'm interested in what happens when we start to try and process these at a larger scale or at a larger concentration. So there are a number of different ways that we can test new mechanophores and polymer mechanochemistry in the lab. The first is using AFM, or atomic force microscopy, and that's where we try to attach a single polymer chain essentially to a tip and pull it away from its surface and see how much force that takes. And that is an incredibly difficult experiment to do. You effectively have to catch a single polymer chain on a really tiny tip and make sure it stays in contact the whole way as you pull it away from the surface and measure the forces that are involved in that. And you have to do a lot of repeats to make sure you have statistically significant results, okay? So it's a difficult experiment, but it gives you incredible information when you think about the mechanophore structure and the forces it needs. Lots and lots of information, but not necessarily something that scales, right? Because we're never really going to be looking at single polymer chain when we use them. The most commonly used technique in the lab is this sonication. So essentially providing ultrasound energy to your solution. It causes these voids to be created in the solution, and as they collapse, they pull the solvent and the polymer chains with them, stretching out the polymer. And we'll have a look at that in a minute. Another way that polymers are really often processed is through extrusion. So essentially melting them, pushing them through a particular shape that we want and extruding out the final product at the other end. Or we can think about simply making some solid material, pulling on them or pushing on them through compression. So I'm going to focus on what happens during sonication and extrusion. And to do that, we've been looking at using this model system, which is polymethyl methacrylate. Acrylates are used in things from nails, not mine, nails, to airplane windows, to display stands at different, in shops and things. Acrylates are all around us, really, really useful. And when we pull on these acrylates, we don't have a mechanophore in them at this point. We break the bond in the center of the polymer chain, and we can follow that by adding a dye that changes color. So we can go from pink to yellow in this case, and we can follow it quantitatively using spectroscopy. So we can measure the absorbance of the solution and see how many of our bonds have broken, or we can measure the length of the polymer chains that we get out at the end. So that's what we do. And what we see is when we look at sonication, this is the process that is going on. And bear with me. Does this play? No, it doesn't. Okay, if I do this. Oh, need to bear with an ad. Is Simon here? Yeah, that's actually no, sorry, we'll go without volume for now. It's okay. So essentially this is a slow-mo video of sonication occurring in a solution. So this tip is vibrating really, really fast and essentially creating these voids. They're bubbles in the solution. And they effectively grow and then collapse. And when they collapse, they pull in the solvent, and that stretches out the polymer chain that's with it. So it's quite a violent process, actually, if you look at it in slow motion. So we're creating this really turbulent flow in the material to look at how this stretches our polymer chains. So when we look at sonication in our system, we see a number of things. We see a dependence on chain length, as we kind of expected. We get, it gets easier and easier to break our polymer chains as they get longer and longer. We see an effect of concentration. So as we increase the amount of polymer in the material, it gets harder to pull the polymers apart. So if you think if we have a really concentrated solution, it's kind of hard to separate these out and pull them and get enough tension across the material to activate them. Whereas if we just had a single chain, it would be kind of easier to pull it out and extend it and get enough tension in the chain. And then, so we get a reduction in the amount of mechanochemistry we see. And then, as we'd expect, if we sonicate it for longer, essentially if it has to experience that really turbulent and violent system for longer, we get an increase in mechanochemistry. So we get more mechanochemistry as we increase the sonication time. So this is the technique that's used in most labs to test new mechanophores to see if we have this polymer mechanochemistry. But the question is it so relevant when we try to think about processing and how these materials are actually going to be deformed when we try to make a product out of them. And to do that, we need to start thinking about what happens to them when we extrude them. So extruding is, like I said, a process of effectively pushing something through a small hole. It's really, that's about as difficult as it is. This video is actually extruding lots of different types of food. So we've got pasta here. We can extrude into lots of different shapes. So we can have, round rods. We can have flat shapes. We can have these very tasty puff-like breakfast materials that are made by extrusion. Other things that are made by extrusion like this are things like Monster Munch and hula hoops are all made by this process, by pushing something through a small die. So this is with food, but we can also think about it for polymers. And this is where I might need the sound so that you don't have to listen to me talk on constantly. No. Anyway, I can talk over this bit. Essentially, it's the same process, but effectively what we do is we heat up the polymer material. So it's in its molten state. It's then forced through what's known as a die or the small opening. And that can be various different shapes. So it can be these rod-like shapes. We can get pipes. We can get various different angles and shapes. So when the molten material is passed through the die, it's then cooled very rapidly and we get these solid materials. We can get flexible, essentially plates. We can get thin wires. We can get all kinds of different shapes. We can get wire coatings. And this is the process that loads and loads of polymer products are made using, okay? So then how does our mechanochemistry in sonication compare to, compared to extrusion? And that's what we've been looking at over the last while. And we found that there's a number of things that are also important that aren't so important when you look at sonication. So viscosity is really important. As we increase the viscosity, we increase the amount of mechanochemistry we get. As we decrease the nozzle diameter, so as we decrease the size of the nozzle that material has to go through, we increase the amount of mechanochemistry that we get. That arrow is pointing the wrong way, or I've said that the wrong way around. As the nozzle gets smaller, we get more mechanochemistry. And then as we increase the flow rate, we get an increase in mechanochemistry. So essentially, as we push it through the hole faster, we get more mechanochemistry. So we can stretch out our polymer chains faster and get more mechanochemistry. And this is all just using PMMA, like I say, an acrylate that's used in all kinds of structural materials all over the place. What happens if we then include a mechanophore? So we started off including a mechanophore that was fluorescent. So we get light given out when it's broken open. And what we see is that we can control effectively the amount of fluorescence or light that we get given out by controlling the number of times we pass it through our nozzle. So if we pass it through our nozzle multiple times, we get more and more fluorescence out of the material. And we've also looked at, this is if we have a single mechanophore in the middle of the polymer chain. But what happens if we move to a system where every repeat unit in the polymer is now a mechanophore? So every single one of these paper clips now has the potential to open and react mechanochemically. And we see that we now get a whole host of mechanophores in the center of the polymer chain are activated. And we go from, in this case, a species that's clear, colorless, to something that's yellow. And this is really exciting because not only is it yellow, it's also conductive. So we can start to think about printing different materials and having something that's insulating and something that's conductive printed from the same starting material. And that's really, really exciting. So we have these differences. We can think that sonication is incredible for telling us about mechanophores and mechanochemistry. We can see that chain length is important, concentration is important, and sonication time is important. But then when we start to move to extrusion, there's some other parameters that we need to think about: viscosity, the nozzle diameter, and the flow rate. So it's both are very interesting. And my argument is that we need to study both at the same time, not just focus on sonication as this method of examining mechanochemistry. And then lastly, I'll leave you with a couple of slides looking at where I think is going to be exciting in the future. And the first thing, essentially coming back to what I've just said there, this idea of how it scales. So we can look at what happens on a single molecular level, what happens with that really difficult AFM experiment, how that scales into something like sonication, where we're looking at dilute solutions. But then really importantly, how the stuff that we learn during sonication then translates into polymer processing and through extrusion. So how do the lessons that we've learned at one scale apply at a further scale? Do we have tolerance in the molecular length, in the molecular weight distribution, in the concentration, and what type of chemistries we use, and the mechanophores that we use? And then that's just, I've mentioned kind of looking at nozzle flow, but there's a whole range of different polymer processing techniques that we can start to examine. So what happens when we look at stuff like electrospinning? What happens if we look at things like slot coating and blade coating? And lots of things are made using these techniques because they're really high throughput. And again, putting a lot of force on a polymer material. So what happens to the bonds that are involved in that? And then finally, what I'm quite excited about is this idea of in situ control. So when we're pulling these polymer chains, they're reacting essentially to a mechanical force. And the thing about processing is that mechanical force is inherent to polymer processing. We can't process the polymer essentially without stretching it at some point. So what if we use those forces, again, not as something destructive where the polymer chain might break, but to enhance their properties? So if we include mechanophores into these materials as we're processing them, can we get out a material that has enhanced properties at the end of processing rather than something that's been broken? And then finally, I think there is this possibility for an impact in terms of their sustainability. So if we think about trying to make a material, if we look at this beautiful logo here, made up of a couple of different parts. So we need a material to make the blue component, we need a material to make the purple component, we need a material to make the yellow component, and then we need to bring them all together to make our final product. But what if we had, similar to what we were talking about in terms of the conducting polymer a couple of minutes ago, where we had the mechanophores, each of the mechanophores giving us a conductive polymer or a conductive portion of the polymer. What if we could then take away the blue component and take away the yellow component? And instead, from one starting material, we could make all of these different final properties from one starting material by varying the force experience during processing. So we could cut down on all of those extra starting materials, we could cut down on all of the extra processing needs, we could cut down on the assembly, and essentially reduce the amount of energy that we need to use this final component at the end of the day. So I think that's quite exciting. And then I will leave you with this, essentially, this one note. Polymers are everywhere. That's what we've said all the way through this talk. And polymer mechanochemistry is not necessarily a bad thing. It's something that's making them stronger, smarter, and more sustainable. So with that, I will like to thank a couple of people, collaborators in Cambridge, in Germany, in California, a whole pile of people who've funded my research. This guy here who's leading the wine tasting is a really good pal who's done lots of the mechanochemistry work with me. Etienne is fantastic. He's done a lot of computational modeling that I haven't gone into this evening. But it's been an absolute pleasure to work with all of these people, and it has been a pleasure to talk to you. So thank you very much. I will leave you with the final question. Is this a good thing or a bad thing? But thank you for listening to me.